Welcome to the first of our background videos for the final projects. The goal of the projects, remember, is to annotate and analyze the TE content of some new chromosome scale genomes. In this context, annotate, or I'll call it annotation as well. It refers to the process of identifying and describing the types and locations of TEs in a genome. The end product of any annotation, whether it's TEs or if you're annotating genes or some other genome feature, usually the end product is a GFF file. We've encountered GFF files at the beginning of the semester a long time ago, learned how to parse them a little bit. But remember, they describe the exact location of each feature in the genome. So for example, where the TE is located, or an exon, or a messenger RNA, if we were studying something else. To get the actual sequences for any given feature, you query the genome sequence file, which is the FASTA file containing the sequence, using the base pair locations from the annotation file, which is the GFF file. So ultimately, that's sort of one of the objectives of our analysis, is to annotate the TEs in these genomes. The rest of this video is going to provide a brief overview of what TEs are, and TEs is just shorthand for transposable elements, or sometimes they're also called transposons. All three of these terms, TE, transposable element, transposons, they're all synonymous. So I'm going to provide a brief overview of what they are, their distribution in the genomes, and that have already been studied, and why we should study them some more. Details about conducting the analysis for the projects are going to be presented in Module 8.2, but I knew you'd probably already be curious about how's this going to work out. So briefly, I'm going to put you into groups, and then each group is going to be assigned a unique genome to analyze. And then Module 8.2 will go through how you do the analysis. Okay, so what are TEs? For intro material, you've got this video, which is, I'm going to provide a brief overview. And then I've got another video where I break down some of the molecular evolution concepts that I think are important to know before you watch these other videos, and so that you can understand all the inferences we're trying to make when we do the annotations and then try to analyze what's going on in these genomes, how these TEs are invading the genomes and when they're invading the genomes. So I've broken that molecular evolution part into its own video. Then the next two provide a little bit more in-depth sort of history of how we know what we know about transposable elements, and then how they work. And rather than listen to me, I've given you two lectures from Dr. Sue Wessler. She is a member of the National Academy of Sciences, one of the premier people working on transposable elements. And even though these videos came out in 2012, so a few of the exact numbers about how many are in a particular genome are not up to date, they provide a great overview and background from a leader in the field. And then finally, I'm also giving you a paper from a few years ago that lists 10 things that you ought to know about transposable elements. All of this content will help you with the quiz material. So if you're watching this video, you should have already taken the pre-quiz, and then once you've covered all and studied all this material, you're using that pre-quiz as a study guide, and then when you're finished with all this, you can take the post-quiz to see how much you've learned. Okay, so what are transposable elements? Basic definition is a DNA sequence that can change its position within the genome. They are considered selfish genetic elements. You can think of them like viruses that live in the genome. There are many types and subtypes of transposable elements, just like there are many types of viruses. They're classified based on their gene content. So over here on the right, you can see a little cartoon representation of some of the different kinds of transposable elements. So I've highlighted in red here just some of the nomenclature that you're going to see recurring in some of the subsequent slides. So we've got the class 1 transposons, which all the class 1 are called retrotransposons because they have, point 3 here, they share a mode of transposition. They all have an RNA intermediate where they have to be transcribed and rewritten from RNA back into DNA to integrate into the genome. Because they're so numerous, especially in eukaryotic genomes, you often see LTRs. LTR stands for long terminal repeat lines and signs. You'll see these broken out as their own pie charts because they're so abundant. But remember, all three of those fall into class 1 retrotransposon. The other main category are the DNA transposons. These do not have an RNA intermediate. They transpose by a different mechanism. And so these are just some terminologies and why things get classified differently, right? So the last sort of subdivision you see is autonomous and non-autonomous. And that's relevant, we'll see in a few minutes when we talk about human genome. Lines, for example, are autonomous elements and signs are not. Lines contain all the genes they need to do their own transposition, whereas signs do not. They don't encode the proteins that they need to move in the genome. Instead, they're actually using the protein machinery of the line elements to move the sign elements. So where are transposable elements in the genome? Most TEs are found in intergenic regions. So this little cartoon, very superficial way of looking at a genome. What we typically consider the functional part is the protein coding sequence and maybe the cis-regulatory elements that govern when and where this protein is going to be expressed. And then in between that functional stuff, we've got these huge intergenic regions. Well, most TEs are found in those intergenic regions. There are a few subtypes of transposable elements that have, I put preference in quotes there. Their mechanisms of transposition bias them to integrate into introns or regulatory elements. So the first thing that really sort of got me interested in transposable elements, because I've for a long time been interested in genome architecture, was this slide. And what it's showing is in the blue bars on the background is the total size of each of these genomes in base pair. So, right, human is roughly 3 gigabases, so 3,000 megabase pairs. Well, if you look at what explains how big a genome is, if you look at the protein coding content, you can see there's hardly any variation in the protein coding part of the genome compared to the tremendous variation in total genome size. Well, then what explains that genome size? It's the TE content, right? There's a really good correspondence between those yellow bars, which is the total amount of TE, and the total genome size. So, right, these things are making up the majority of variation in genome size. So really interesting in terms of, that seems weird, right? They're these selfish sort of parasitic elements that are blowing up genomes. If you look at the human genome specifically, somewhere between 45 and 50% of our genome is just transposable element content. So here, like I said earlier, you often see these broken down into subcategories, right? So lines, signs, and LTRs, these are all retrotransposons, so they have an R RNA intermediate. They are the copy and paste, is another term that you'll see used for retrotransposons. DNA transposons, which are cut and paste mechanisms, they don't comprise such a huge amount in our genomes. But you can see, like, compared to how much is protein coding, what we traditionally call functional, which is only like 1.5 to 2% of our genome, huge amount. And so questions arise about, well, is it an important amount? How do we get away with having all this stuff that could be jumping around in our genomes? If you look across the human genome, the composition in terms of what types of transposons is pretty consistent across the chromosomes, right? You see, like, these proportions are sort of similar. And in human genomes, right, the three biggest chunks here, the blue, the red, and the green, are all retrotransposons. Okay. So a huge proportion of the TE content is due to these retrotransposons. And in our genomes, in particular, the line elements and the sign elements are what's taking up, 30+% if you combine those two. That's not, that sort of pattern of high retrotransposon content is not universal. So here I'm showing you the TE composition of pygmy mole crickets, just one that I had handy. But the TE composition varies a lot, and it can vary a lot even between closely related things. I didn't have to pull something that was so far away as a cricket evolutionarily, but it demonstrates the point that, remember, our genome, just this little yellow sliver, that's the DNA transposon content, compared to these chromosomes in pygmy mole cricket, where, like 50% or more of the transposon content is due to DNA elements. So very different transposable element landscape going on in these crickets. And like I said, that can be highly variable even between closely related things. Going back to the human genome, I just wanted to highlight one of the things. You can't talk about the human genome and human genome variation for very long without talking about what we call ALU elements. These are a 300 base pair long subfamily of sign elements, right? So these are a non-autonomous retrotransposon. It's the most common transposable element in the human genome by copy number. And I highlight underline copy number here because it's not the most common by base pairs because these are such short elements. But somewhere like 10% of our genome is just these ALU elements. I've seen numbers recently where it's estimated that there may be more than 1 million copies. It's certainly somewhere between 500,000 and 1 million copies of this thing in our genome. So, like, this is a little piece of genomic virus that has just gone crazy in our genomes. The transposon that is the most common by base pair is the L1, and this is a type of line element, right? So that's the autonomous version of this retrotransposon. And so the ALU elements are using the L1 machinery. The reason there's more base pairs in L1 is that it's just a bigger transposon. It has all the machinery. It's encoding the reverse transcriptase and all those other genes that both kinds of elements need in order to mobilize in the genome. Between them, right, they're explaining 25 to 30% of our genome. Just crazy. This study, that the ALU repeat in human genome diversity, they went and looked phylogenetically when we started to get some genomes of other primates. And it's just interesting to note that there have been sort of unique explosions of these things that at various times in primate evolution have just gotten loose in our genome and started to amplify. In the other lecture on molecular evolution, we'll talk about how we can trace this phylogenetically. But note, there are families of ALU elements that are unique to our lineage, and it is ongoing expansion and mobilization of these elements today. Any two individuals, I don't remember the number off the top of my head, but differ by several ALU positional variations. Last note here on sort of the history of what we know and what are transposons. They're not all bad. So I wanted to make a point to say there is something called a transposon domestication, where this is when TEs adapt to function in the cellular process of the host organism. There's a whole really interesting literature on this. They tend to be recruited as transcription factors because many transposons recognize themselves as targets for the transposon machinery. So they're sort of seeding their own binding sites all over the genome. And so if they're located in regulatory elements for a protein coding gene, then they have the ability to go and either cut that regulatory site or modify the expression of that gene in some way. And so we think, and there's good evidence, for example, in rewiring the regulatory network that's involved in placenta formation in mammals, that many of the genes that are involved in placentation have transposable elements in the upstream region, and they're all governed by a transcription factor that looks very similar to an ancestral transposable element. And then it's a very similar story for neuronal genes. So maybe even affecting big brains or complex neuronal networks in human evolution. So this gets us into why do more of this? Why is this an interesting and a real research project that you're going to be involved in? Well, the number of sequenced genomes has increased exponentially over the last 10 to 15 years because of cheap short read sequencing. Most of that short reads is almost synonymous with Illumina, which is the sort of brand. We talked about that in previous modules. So lots of genomes out there based on short read sequencing. And so these two graphics show transposable element annotations and study based on those short read genomes. And you can see some of the things that I talked about before. There's lots of variation across species in the TE content. There's variation in the percent of the TEs in the genome. So this top one is trying to highlight, the percent of the TEs. So some of them are over 50%. Some things like Fugu here, less than 10% of the genome is TEs. So it's interesting why some big genomes have lots of TEs and others seem resistant to them, right? It's like some have some you could think of it almost like immunity to having these TEs get loose in their genomes. And then the bottom one here is just highlighting the relative proportions of those things. So how much variation in the subtypes of TEs are in those genomes. And then the one on the right is the same thing, but for invertebrates. So again, lots of variation, both in the proportion of the genome and the types of TEs. So historically, and again, just to highlight, these are all based on short read sequencing. So and the genome assemblies that were based on that short read sequencing. A consequence of short reads is that you get highly fragmented, in other words, poorly assembled genomes, because there's an inability to assemble the repetitive DNA. So the transposons in particular are poorly assembled in all of those short read sequence genomes. And if you have high transposable element content, it leads to a bad assembly, which then leads to a bad TE annotation. And so this cartoon at the bottom is to sort of try to unpack that a little bit. Why is that? So this first line is the actual genome. So if you were going to go into the organism, you have perfect knowledge. This is the actual a segment of the genome where we've got some unique sequence over here, and then we've got 4 transposable element copies from the same type of transposon, and then a bunch of other unique sequence. Well, when you do your shotgun short read sequencing, you can line that up to the genome, or sorry, not line it up to the genome. You can line it up to each other. And so you're basically lining all these short reads up, looking for the overlaps among the short reads. When there's unique sequence, that's not that difficult, right? Because they only overlap in one way. There's only one case of that in the genome. But when you get to repetitive content, what happens is, like, if you were trying to align this read up to the genome, it goes in 4 different places. Now, when you're trying to assemble it, it doesn't know that there are 4 instances of this thing in the genome. So it doesn't really know how to deal with this. And it's going to just stack these up as though they were unique. So even in the best case scenario, what will happen is you'll get an assembled genome that's contiguous, right? We got one stretch that goes through this whole chunk of the genome, but we only assembled one copy of that transposable element, right? Because all of these things, wherever there's overlap, they're all going to assemble just one copy of this thing. Now, you might, because there are 4 versions of it, you might have a lot of reads. And so one of the bioinformatics ways people have historically tried to deal with this is look at the read depth, because you should have like 4 times the number of reads coming from these orange things because there are 4 copies of them. And so like in this assembled genome, you have 4 times the number of things that correspond to that chunk than would correspond to the upstream or the downstream unique regions. But what often happened is that if those transposable elements are scattered all over the genome, the assembly just can't put it together because it gets a few reads that are like hanging off the edge here. And so it looks like it belongs next to that, or this end looks like it belongs next to that. But if it's scattered around the genome, you have lots of different positional information. And so it just doesn't stick these 2 things together. It leaves them broken into different, we call them scaffolds. So the result then is you get these genome assemblies. And in an organism with 10 chromosomes, say, you can have an assembly that has thousands of scaffolds, right? So I think naively, if you're not used to the way short read genomes are, you think of an assembled genome as having roughly the same number of scaffolds as it has chromosomes. That is not the case at all with short read Illumina genomes. Typically, you have thousands, maybe tens of thousands of scaffolds, and it's all because of the difficulty of short reads to assemble repetitive content. In the last 5 years, long read sequencing from PacBio, Oxford Nanopore, talked about these in previous modules, has become more cost effective and resulted in a really very rapidly growing number of what I quote unquote good assemblies. So that's what I'm calling chromosome scale assemblies, because individual sequencing reads from PacBio and Oxford Nanopore can span multiple repeats. So again, we've got our actual genome at the top there. And then when you have these long reads that are, say, 20,000 base pairs long, it actually spans from the unique sequence, either, if not all the way through, at least this read alone tells you that there are at least 4 copies of this transposon in this region. And then you get other reads that sort of confirm the location of the other end. And now you can basically assemble the actual chromosome scale right through the number of repeats and have a much better reflection of the actual TE content. You can tell these are better genomes because, or good genomes, because the number of scaffolds in these genomes are close to the number of chromosomes in the organism. Okay. So given that we're getting all these new genomes coming online that are chromosome scale, it's a prime time to go back and look at what is the TE content in these things. So final projects, we're going to describe the TE content of new chromosome scale genomes. This is the first module where we're understanding the fundamentals of TE invasion. Second one, we'll talk about their molecular evolution. Hopefully you've already taken the pre-evaluation. You'll do the intro lectures, PowerPoint reading, do the post-evaluation. Okay, so you're going to retake that test. And then in Module 2, we're going to learn to run Earl Grey, which is a state-of-the-art software pipeline for TE identification. Watch some videos that talk about the challenges of annotating TEs, and hopefully find some interesting stuff about the variation in transposon content across some really good genomes. Okay. So next thing for you is to go ahead and watch the molecular evolution video.